All right, so this is the second lecture for Chapter 4. In the previous lecture, we talked about protein structure and protein function. Now in this lecture, we're going to talk about protein synthesis. So again, just to come back to this figure that we've visited throughout these lectures, you start with DNA, then you get transcription. We've talked about forming our mRNA, and then we're going to talk about this last leg here, translation. So to talk about translation, you got to talk about the genetic code. So the genetic code consists of triplet sequences of nucleotides that are called codons. So we come down here to a sample mRNA. So this sequence of 3 nucleotides, this is a codon, this is a codon, and each of these codons code for a specific amino acid. So this codes for an amino acid, this codes for an amino acid, and then so on and so on. So each of these will code for a specific amino acid. And then these are all bound together by that peptide bond to form the polypeptide chain. So to decipher the genetic code, this chart comes in handy. You've probably seen this before. This has all the codons and then their corresponding amino acids. And we'll go through how to read this chart on the next slide. One thing to note is that each codon pairs with an anticodon on a segment of tRNA. So if we have a codon, so we'll say this first one here, U, and obviously there's going to be nucleotides on either side of it. And this will be our mRNA. There's going to be an anticodon, which will be, in this case, AAA, and this will be part of the tRNA sequence. And then the tRNA is carrying the amino acid that corresponds to this codon. So in this case, it would be phenylalanine. Another thing we want to point out here is the start codon, AUG, which corresponds with methionine. So methionine is actually the first amino acid in every polypeptide sequence because it corresponds to the start codon. And the start codon is where polypeptide synthesis begins. So once this sequence is recognized on the mRNA, that's where the ribosome then begins translation. Now there's only one start codon, but there's actually 3 stop codons, UAA, UAG, and then UGA. And these 3 signal to end translation. So you've reached the end of that polypeptide sequence, and then translation is over for that particular sequence. And as we say here, start and stop codons flank the sequence of codons that code for a polypeptide. So you have your start codon, and then you're going to have, C1, C2, meaning codon, and then eventually you'll have a stop codon. And now we'll go through an example on the next slide. So here we have an mRNA sequence. We're going to go through this using the chart here. So we'll start with the first codon, which, as you may have guessed, is the start codon, AUG. So you have methionine here. So we'll start with methionine. Then we go through ACA. So we start here with the first letter, as you can see here. Then the second letter, C. So we come down here, and you have ACA, which then corresponds to threonine. So that's our second amino acid. Then you have UAC. So UAC is here. So U, A, and then you come down here, UAC. So that's going to be tyrosine. Then you have AGU. So we start here, A, G, come down here, AGU. That's going to be serine. Then we get to the last codon here, UGA. So again, we go U, G, A. And what do you know? That's a stop codon. So this is where it'll stop. And so this is our final polypeptide sequence. Now in real life, they would probably be much longer than this, but this is just to demonstrate how this process is carried out. So a key player in the process of translation is transfer RNAs or tRNAs. Now here's a structure of a tRNA. And as you can see, it's a sequence of nucleotides, just like an mRNA, arranged from 5' to 3'. And you can see it has some base pairing that forms these loop-like structures. And what you want to pay attention to is the anticodon loop. So we've talked a lot about this already. This sequence is what base pairs with the codon sequence in the mRNA. So let's draw that out here. So we have mRNA sequence here. We're going to do 5'. We'll start 5' over here because here's the 3' here, 5' here. So on the tRNA, this is 3', this is 5'. So we have a few filler nucleotides like this. And then we'll have U, C, and then a few more like this. So UUC is the codon, okay? And it's base pairing with the anticodon like this. Now this codon actually codes for phenylalanine. So this tRNA is going to be carrying the phenylalanine that corresponds to this codon. So that's how so essentially this is how this process works, is the tRNA comes in, its anticodon base pairs with the codon in the mRNA sequence, and the tRNA is carrying the amino acid that directly corresponds to that codon. So that's essentially how the genetic code is translated. So there's a few features of the genetic code that are important to remember. First, it's universal. So what does that mean? It means that it's the same code for codons to amino acids that's used in protein synthesis across all of human biology. So in all tissues, this is how protein synthesis occurs, and this is the code that it follows. The only minor exception to be aware of is mitochondria, and that's because it has mitochondrial DNA as well. It's non-overlapping and commaless. So what does that mean? All nucleotides in a translated mRNA strand, so the strand that we're not talking about the pre-mRNA that hasn't undergone processing yet. We're talking about the final mRNA strand that makes it out of the nucleus and attaches to the ribosome. All of those nucleotides in that strand are part of a codon, and no nucleotide codes for an empty space or part of more than one codon. So what does that mean? So if we have a sequence here, we'll start with AUG because that's our start codon. This is an mRNA sequence. And then we'll just make some up here. CUG, AUU. So each of these are a codon, right? So it's 3 sequences. Each of these code for one amino acid. None of these nucleotides are going to be part of more than one codon. They're only part, so this G is only part of this codon. It's not part of this codon. Same thing with this G here is only part of this codon. It's not part of this one. The other thing is that all of these nucleotides are part of a codon. There's no single nucleotide or group of nucleotides that are hanging out where they don't code for anything. Essentially, as we say here, empty space. So bottom line, every nucleotide is part of a codon, and it's only part of one codon. It's unambiguous, so a codon only translates to one amino acid. So again, you have AUG, CCA. So each of these, it's a 1-to-1 ratio. So it's only going to code for one. So this is only going to code for methionine. This is only going to code for, let's see here, CCA, proline, okay? This is only going to code for a proline. It's not going to code for more than one amino acid. So it's one codon for one amino acid. Then degenerate. So you have a few amino acids that can actually be translated from more than one codon due to the wobble, which we'll talk about in the next slide. So just to differentiate this statement from this one, this is describing what's happening during the actual process of translation, is that when you're translating a codon, that codon only translates to one amino acid. It does not translate to any more than that. This is talking about the code itself. So if you look down here, so leucine's a good example. Leucine has multiple codons that all code for it. So each of these codons, they all code for leucine. Same thing here, proline, threonine, all of these amino acids here, they can be coded for by more than one codon, and that's due to the wobble effect. So the wobble position on a codon, it specifically refers to the third nucleotide in the codon sequence. And so if we come down here, you have U, U, C, and that's our codon. So 5 prime, 3 prime here. This is our codon, and it codes for phenylalanine. So this tRNA will be carrying phenylalanine like we talked about. So the base pairing that occurs between these two adenines and these two uracils is much stronger than the base pairing that occurs from this guanine and cytosine. So what that allows for is actually several non-Watson-Crick base pairing to occur. And so what that results in is that many codons with the same first two nucleotides and different third nucleotide still code for the same amino acid. Again, a good example here is leucine. So you can see here, CU, 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 and so on. They're all the same first two nucleotides. However, the third nucleotide is different in all of these codons, but they still all code for leucine. Same thing here with valine. All these are GU, 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 but then they all have a different third nucleotide, but they all code for valine. And that's because of this wobble, and that's because this third nucleotide base pair is not as strong as these first two. Now, each tRNA that's out in the cell is actually specific for one amino acid. But before that tRNA or any tRNA can take part in translation, they have to be charged, which means they have their corresponding amino acid added to them by an enzyme called aminoacyl tRNA synthetase. Each type of amino acid has its own aminoacyl tRNA synthetase. So here we have phenylalanine, which corresponds to this anticodon. You need to get this phenylalanine onto this tRNA so that it can become charged, right? And then be primed and ready for translation. So you have phenylalanine aminoacyl tRNA synthetase, which recognizes phenylalanine, it recognizes the tRNA and brings them together. So it'll catalyze the addition of this amino acid to the tRNA, just like we show here. And so now it's charged and ready to be a part of translation. Now, one interesting thing to note is that aminoacyl tRNA synthetase has proofreading ability to detect and remove amino acids that are attached to the wrong tRNA. So just another mechanism of protection to assure the genetic code from gene all the way up to protein is translated with the highest integrity. So protein synthesis overall, so the process of translation takes place at the ribosome, like we've talked about, in the cytosol, and it has 3 phases: initiation, chain elongation, and termination. So if we draw a mRNA sequence like this, okay, this is mRNA. And then we have the ribosome that comes in like this. So it's going to start at the start codon. So this would be AUG. And you're going to have the tRNA that corresponds to that. Bringing methionine in. So that's going to come in. Its anticodon is going to complementary base pair with this AUG. And that's going to start the process of methionine. So that's what's called initiation. And because of this, the initial amino acid in all polypeptide sequences is methionine. And that's because the start codon for all of these corresponds to methionine. Then you have chain elongation, which is where the ribosome is then going to go down the mRNA sequence, and more tRNAs are going to come in corresponding to the codons that it encounters, and then you're going to start generating a polypeptide chain with these peptide bonds between amino acids. Then it's going to get down here to the end of the sequence where it's going to hit a stop codon. And then when it gets to that point, the ribosome will fall off, and you'll have your polypeptide chain. And remember, there's 3 different stop codons. So any one of these will trigger termination of translation. So the process of translation is carried out by the ribosome in the cytosol of the cell. And so one of the first steps of translation is assembly of the ribosome. And so ribosomes are assembled from 2 subunits, the large and the small. So you have the small subunit, which is known as 40S in eukaryotes, and then 30S in prokaryotes. And this is what actually binds to the mRNA strand. So here's our mRNA, 3' in down here. And then it also binds to the aminoacyl tRNA that's coming in. So ribosome is moving down, so you'll have a codon piece here. So then you'll have the tRNA here carrying the amino acid that corresponds to the codon right here. So then this will come in, and this will bind to the 40S subunit. Now the large subunit, which is out here, this is known as 60S in eukaryotes, 50S in prokaryotes. This will bind to the small subunit after the start codon has been bound. So after this tRNA carrying methionine has bound to the start codon, so first 40S, the small subunit, binds to the mRNA. Then the tRNA carrying methionine comes in and binds to the start codon, which is AUG. Then lastly, third, the large subunit will come in and bind to this complex. And what's important to know is also that the large subunit has what's called peptidyl transferase activity, and this catalyzes the formation of those peptide bonds. So you'll start with methionine, and then you'll have more tRNAs coming in as the process of translation occurs, and so you'll have new amino acids coming in, and so you need to form that peptide bond. And so you keep adding to the chain, and then you'll keep adding, and that forms your polypeptide sequence. So this initial process of the ribosome assembling and binding to mRNA and the methionine tRNA coming in, this is what's called initiation. So this is the first part of the actual translation process. And again, this begins with that binding of the small ribosomal subunit to the mRNA strand and the start codon, and then the methionine tRNA. And this forms a complex called the 40S initiation complex. So then you have initiation factors, which are proteins that bind to the mRNA. They're analogous to transcription factors, during transcription. So they bind to the mRNA and they help facilitate formation of this initiation complex, and also help facilitate recruitment of that methionine tRNA to begin translation. So then the large subunit comes in and binds, like we said, and that forms the ADS initiation complex, which consists of both of these subunits and then the mRNA and then the start codon bound to the methionine tRNA. Then the next step is elongation. So here we'll draw this up here. You have initiation, which we talked about on the previous two slides. Then you have elongation, which is the process of actually moving down and carrying out translation of the rest of the strand. And then you have termination, which is when you hit those stop codons. So elongation, it proceeds by translating from one codon to the next. So here you have your start codon here, which is AUG, which corresponds to methionine. And let's say you have the next codon is CAU, and then after that is GUA. So elongation is, so initiation is getting to this point here, and then elongation is going from codon to codon to codon, and then you eventually will get to the stop codon, and that's where you hit termination. And this occurs in the 3' to 5' direction. Remember, these conventions are really important. So again, here 5' to 3'. So in this figure, translation is occurring in this direction. Translation begins at the amino end. We've already mentioned this. This would be the N terminus. So this first amino acid here is methionine, right? So this would be your N terminus. And then if this were this is the most recently added amino acid here. So if this were the last one added, let's say, you know, the next one it hits a stop codon. If this were the last one added, this would be the C terminus, or the carboxyl end. So just like how you had initiation factors during initiation, you have elongation factors during elongation. And so these are also proteins that come in and bind, and they help with two things, really helping move to the next codon and then receive the incoming amino acyl tRNA that corresponds to the next codon. So here, this would be the next codon. Here, you have this tRNA that's bound. So this tRNA would have came in from this direction, and you have the anticodon here, which would have base paired with it. So it helps facilitate that interaction. So it just really, again, helps facilitate the process and keep things moving efficiently. And so when this comes in with the corresponding amino acid to the next codon, this amino acid then needs to get added to the growing chain here. As you can see, we have this growing polypeptide chain. So you're going to form one of those peptide bonds. And so a peptide bond needs to be formed between the last amino acid that was added and the next one where corresponding to the next codon. And this peptide bond formation is catalyzed by the ribosome component of the rRNA of the large ribosomal subunit. And this is the same thing as what we talked about earlier, peptidyl transferase. So this terminology, both in this bullet point and then when we talk about peptidyl transferase, is referring to the same activity, the same concept. All right, so just like there were initiation factors for the initiation process, there's elongation factors, or EF, for the elongation process. And one function they have is to help facilitate movement of the ribosome down the mRNA towards that 3' end, helping it move from codon to codon. And the next thing they do is they help receive the next tRNA, which corresponds to the next codon that is encountered on the mRNA strand. So you have this amino acid here connected to this tRNA, and this was the last added amino acid to this growing polypeptide chain. So then the next tRNA that's carrying the amino acid that corresponds to this codon here, that's the next codon, this tRNA has to come in and bring that amino acid in, just like that. And so elongation factors help with that process as well. So now that this tRNA has brought the next amino acid in, the next step is to form the peptide bond. And so a peptide bond is formed between the last added amino acid to the polypeptide chain and then this new amino acid that's brought in. So we'll label that PB for peptide bond. And this is catalyzed by the enzyme peptidyl transferase. Peptidyl transferase. And remember, this is part of the ribosomal RNA of the large ribosomal subunit. And so it's also called a ribozyme. Now, a ribozyme is just a generic term that refers to RNA molecules that carry out enzymatic functions. And so that's what peptidyl transferase is. It's not an enzyme per se, it's a ribozyme. and it's a component of the large subunits rRNA. So just to summarize, this tRNA brings in the new amino acid corresponding to the next codon. This peptide bond is formed between the last added amino acid and the new amino acid, thus adding this new amino acid to the growing polypeptide chain. So over the next few slides, we're going to talk about these 3 sites on the ribosome: the A site, the P site, and the E site. And these are critical for helping the process of translation move along efficiently. And they particularly are important for receiving the tRNAs carrying the amino acids that are corresponding to the next codon that's encountered by the ribosome on the mRNA, and then facilitating the addition of these amino acids to the growing polypeptide chain. So that first site, the A site or the aminoacyl site, this guy right here, is initially empty when the ribosome moves down the mRNA and encounters the next codon, which would be here. And then it's ready to receive the next aminoacyl tRNA or charged tRNA carrying the amino acid that corresponds to this codon. So this guy comes in just like that. And now this tRNA is in place. And remember, it's base pairing with this codon here that corresponds to this amino acid, which is the next in line to be added to the polypeptide sequence. So then the next step is, once this tRNA is brought in the amino acid, is then you form a peptide bond between the last added amino acid, which would be this guy here, and this new amino acid brought in by the tRNA at the A site. And remember, this is catalyzed by peptidyl transferase. So it makes sense that the P site refers to the peptidyl site. And the P site contains the tRNA that's holding the growing polypeptide chain. And that makes sense because as these amino acids get added, the polypeptide chain then gets transferred to the next incoming tRNA, which would be this tRNA in this case. So what's going to happen is that after this peptide bond is formed, the growing polypeptide chain will get transferred from this tRNA to this tRNA, which then frees up this tRNA to then move down to the exit site. And then this tRNA holding the polypeptide chain can then move to the P site just like that. So after the polypeptide chain has been transferred from this tRNA to that tRNA, and then this tRNA carrying the polypeptide chain has moved into the P site from the A site, this empty tRNA is then in the exit site where then it can exit the ribosome. And then in the meantime, this A site is opened up, and then the ribosome has moved down to the next codon, which then corresponds to this amino acid bound to the next tRNA coming in. And so then this empty tRNA has left, and this aminoacyl tRNA has then come into the A site, carrying the amino acid corresponding to the next codon that's then going to be added to the polypeptide chain. All right, so we talked about the different components of the ribosome and the different phases of translation. So now we're going to just have a quick summary over the next few slides to tie it all together. So you have the ribosome here. It encounters the next codon as it's moving down the mRNA in the 3' direction. The empty A site is ready to receive the next tRNA that has the amino acid that corresponds to this codon. So the tRNA comes in. Remember, its anticodon sequence base pairs with the codon sequence that corresponds to this amino acid. Then you have the peptide bond formation between the recently added amino acid and this amino acid. Then you transfer the polypeptide chain as a result of that, because you've formed that peptide bond, over to the tRNA on the A site from the tRNA on the P site. So now this tRNA is empty and it's ready to move to the exit site, and then the tRNA carrying the polypeptide chain is ready to move into the P site just like that. So now the A site is empty, and then in the interim, the ribosome has moved down to the next codon, which corresponds to this amino acid being brought in by the next charged tRNA. This empty tRNA is now in the exit site after it's passed on the polypeptide chain, and it's ready to exit. And so it exits like this, and then this tRNA carrying this amino acid has come in with this amino acid corresponding to this codon. Same thing again, peptide bond formation, and then you transfer the polypeptide chain over to this tRNA from the tRNA at the P site. tRNA at the P site moves to the exit site. tRNA from the A site carrying the polypeptide chain has moved to the P site, and then again the process repeats. You've moved, in the interim, the ribosome has moved down closer to the 3' end, and now the empty A site can receive the tRNA carrying the amino acid corresponding to the next codon. And then one more time, we'll just show you this process again as one short animation so you can really see it in action. And then that process repeats until the ribosome comes down and encounters a stop codon. And then when it reaches the stop codon, this is the termination phase. So it reaches one of these stop codons, either UAG, UGA, or UAA. So if we draw out 5 prime, 3 prime, this is an mRNA. And let's just say the stop codon here is UAG, and then you have your ribosome like this. So once it reaches this stop codon, there's what's called release factors that come in. And these are analogous to the elongation factors during elongation and the initiation factors during initiation. They come in and bind the translation complex, and they help release the finished polypeptide, because remember there's a polypeptide chain that's been being formed. It helps release that from the ribosome. It allows that last tRNA to come out. And then you have final dissociation of the ribosomal subunits from the mRNA. So you have the small and the large subunit that fall off of the mRNA. And so then you have the free mRNA and the free polypeptide. So again, protein synthesis occurs in the cytosol at the ribosome. And for some proteins, that's only the initial phase. For these proteins, the second phase or final phase is in the rough endoplasmic reticulum, or the rough ER. 3 types of proteins that this mainly applies to are cell membrane proteins, secreted proteins, hydrolytic enzymes utilized by lysosomes. So if we draw a cell here, this is our membrane. We're going to have a ribosome here. Here's another ribosome, and then we have an mRNA being read here, and then you have a polypeptide chain like this being produced. So if it's a cytosolic protein that's going to function within the cell, that's fine. You just produce it, it's in the cytosol, it's ready to go. However, if it's a cell membrane protein, such as a cell surface receptor like this, that's going to bind a signaling molecule, you can't just produce it into the cytosol and then expect it to embed itself in the membrane. Unfortunately, it doesn't just work like that. What ends up happening is it has to go to the rough ER. Remember, the rough ER is usually right next to the nucleus. And so what happens is this ribosome carrying the mRNA and then the growing polypeptide will then translocate over to the rough ER and dock at the membrane of the rough ER, and then finish translation and then release the polypeptide into the lumen of the rough ER. From there, it's going to undergo processing in the rough ER and then be exported to the Golgi apparatus, where it also undergoes processing, and then it's secreted in vesicles that will carry the polypeptide to its final destination. So in this case, let's say it was a cell surface receptor like this that's going to be embedded in the membrane. It's going to be within this vesicle. The vesicle will then fuse with the cellular membrane, and then you have the cell surface receptor embedded. Let's say it's a secreted protein, so such as a endocrine gland, cell in an endocrine gland producing a hormone for secretion. Again, the purpose of secreting it is to secrete it into the bloodstream so that it can carry out its effects. Doesn't do any good just staying in the cytosol. So you secrete the polypeptide hormone here. The same thing, it's in the rough ER, then it makes it into the Golgi, undergoes processing there. Then you have a vesicle that contains it like this. That vesicle fuses with the cell membrane, and then the hormone is released into the bloodstream, where then it can carry on its effects. Lastly, hydrolytic enzymes utilized by lysosomes. So lysosomes, as you recall, are organelles that essentially degrade the garbage of the cell, or broken down proteins or broken down cellular components. And part of the way they do that is by containing hydrolytic enzymes. So they're membrane-bound organelles like this, and they have these hydrolytic enzymes like that in here. Now, to get these here, you can't have hydrolytic enzymes just floating around in the cytosol, otherwise it's going to cause massive damage to the cell. So again, the ribosome is going to come over to the rough ER, release those hydrolytic enzymes, where they'll undergo processing within the rough ER, then they'll make it up to the Golgi, and then they'll also be released into vesicles. However, these vesicles won't go fuse with the membrane, they'll go and fuse with the lysosome, and that's how the lysosome ends up getting these hydrolytic enzymes. So as you can see, there's a method to this madness. The reason it's not just random proteins that are processed in the rough ER. It's to serve a specific purpose because they need to be sent somewhere by the rough ER and the Golgi. So the nitty-gritty details of this, at the terminal end of these polypeptide sequence, so here we have a ribosome, it's in the cytosol, and then you have your polypeptide being produced like that. There's a signal sequence that binds a protein called the signal recognition particle. So it's going to bind the actual polypeptide sequence, and you have it here. So this will be your SRP. What the SRP does is then traffics it to the rough ER membrane, and then it will bind to the receptor, which is the signal recognition particle receptor, or the SRP receptor. And you see that here. Here's the SRP. It's binding its receptor, and that helps essentially dock it or bring it into the membrane of the rough ER. After it binds the receptor, the SRP then dissociates, and then the ribosome carrying the mRNA, shown here in red, and then its growing polypeptide, then docks with the translocon, which is a protein embedded in the rough ER membrane, which then allows for translation to continue, and then the polypeptide is released into the lumen, as you can see here, of the rough ER, where then it can undergo further processing and then go to the Golgi for its final transport. All right, so that wraps up our discussion of protein synthesis.